The present video is regarding the comparison between nitrosamines and nitrosamine drug substance related impurities, that is NDSRIs. Welcome dear viewers to this short video on the comparison, and after watching this video you will clearly understand the differences between these type of nitrosamine impurities. Many of the times we get confused between the nitrosamine and the NDSRIs. So this video will clear your all the doubts and will give you the clarity between these two type of nitrosamines. Nitrosamine impurities are the potent impurities, carcinogenic in nature. That's why these have very stringent limits in the pharmaceuticals. I have made many of the videos on these impurities. I request you to go through the video available on this channel, and this video is for the comparison purpose. So by definition, nitrosamines are the small nitrosamine impurities. These may be many times volatile or semi-volatile nitrosamine compounds. Sometimes these may not be volatile. And NDSRIs on the other hand are the nitrosamine impurities which are structurally related to the drug substance. So as the name indicates, these are the nitrosamines impurities related to the structure of the drugs. Structure of nitrosamines: typically the nitrosamines have simple secondary or tertiary amine with the nitroso group, and NDSRI's structure is complex, containing the drug moiety plus the nitrosamine functionality. Then source of the nitrosamine. Nitrosamine impurities arise from the excipients, reagents, solvents, degradation, or by the cross contamination, while the NDSRI impurities are formed when the drug substance or intermediate contain the secondary or tertiary amines. So one thing should be clear to you, that nitrosamines are the small impurities and NDSRIs are the bigger structure impurities. You can remember like that, and these are related to the API structure. The examples of nitrosamines are NDMA, NDEA, NMBA, DIPNA, EIPNA. So many examples you can find. For NDSRIs, the nitroso group is attached to the APIs. So nitroso analog of the API like valsartan, losartan, rifampin, and many others. Many APIs with the secondary or tertiary amine group can form the NDSRI impurities. Then nitrosamines are generally, or these are often volatile in nature, and NDSRIs are non-volatile due to the large molecular size. Then toxicity concern of the nitrosamines. These are with known toxicity, potential genotoxic and carcinogenic impurities, while NDSRIs may also be genotoxic, carcinogenic, but the toxicity varies with the structure. Risk assessment for the nitrosamine is required and mandatory for all the products. These are controlled via the ICH M7 guideline and the health authority guidelines. Guidances are provided for the nitrosamines and NDSRIs by the US FDA, EMA, ANVISA, Swissmedic, and many other country specific regulatory bodies. NDSRIs require the cross check, and it requires case specific risk assessment and toxicological evaluation. So if the NDSRI risk is not there, then it depends on the case by case basis whether the molecule is sensitive for the formation of NDSRI. Based on that, we can go ahead. Then acceptable intake for the nitrosamine, it is well established for many of the nitrosamines like NDMA, it is 96 nanogram per day. While for the NDSRIs, acceptable intake limits needs to be derived using the structure activity relationship or read across approach. Many of the times SAR activities are performed, read across activities are performed to check the potential of carcinogenicity and to derive the AI limit for NDSRIs. Then for nitrosamine, there are analytical challenges. So for nitrosamines, these are detectable using the sensitive GCMS, LCMS methods, while the NDSRI impurities are difficult to detect due to their complex structure, and this complex nature requires the specific methods like LC, HRMS and some other techniques. For nitrosamines, the guidelines are there. The nitrosamines are covered in EMA, USFDA, ICH M7 guideline and also the ICH Q3A guideline. NDSRIs are included in the recent EMA and FDA guidelines, and these are evolving case by case approach. Many of the times you can find the interim limits for the NDSRIs. Control strategy for nitrosamines is to avoid the precursors, improve the process controls and packaging review. While for the NDSRIs, these involve the deep understanding of API structure, synthetic route and the nitrosation potential. Examples of the drugs for nitrosamine presence and NDSRI presence are the ranitidine, valsat and metformin. These are sensitive molecules for nitrosamines, while the rifampin and sitagliptin are sensitive for the NDSRI formation. Many of the times you can find such molecules which are sensitive for the small nitrosamines and also sensitive for the NDSRI formation, while sometimes you will found the molecules are sensitive or formulations are sensitive for the only nitrosamine formation or only NDSRI formation. But risk assessment is required for both the types of nitrosamines. For NDSRIs, you can get the limit based on the structure activity relationship approach and read across approach. Many of the times the USFDA and EMA, these regulatory bodies are providing the limits for the NDSRIs. So whenever you come across a situation where there are chances of the nitrosamine formation and the NDSRI formation, the total impurity intake limit specification will be very tricky. This video has provided the information regarding the comparison or differences between the nitrosamines and the NDSRIs. I hope this video will help you to understand these two impurities in detail. Thank you for watching the video.